Mitosis consists of 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase, which is followed by cytokinesis. The purpose of mitosis is to produce 2 new, genetically identical daughter cells, and is performed by all somatic cells in an organism. Mitosis is essential for development and healing in multicellular organisms. Interphase is the longest period of the cell cycle. It consists of 3 phases: G1, S, and G2. During G1 phase, the cell undergoes normal cellular activities. If given the signal, the cell will enter S phase, where the cell synthesizes its DNA. After S phase, the cell will enter G2 phase, where it will complete its preparation for division and generally enter the mitotic cell cycle. Prophase and prometaphase are the first stages of mitosis. Here, the extended molecule of DNA begins to condense into chromosomes. A chromosome consists of 2 highly condensed strands of identical DNA called sister chromatids, which are bound together at the centromere. At this point, the cell is diploid and will remain so throughout the mitotic cell cycle. By the end of prophase, the nuclear envelope has completely dissolved, leaving the chromosomes floating within the cytoplasm. Once the chromosomes are free of the nuclear envelope, the cell enters metaphase. During this phase, the cell produces a structure known as the mitotic spindle, using tubulin that is contained in the cytoplasm to create microtubules. The spindles form from either end of the cell around the centromeres. Some microtubules connect to the chromatids at the connecticores, protein structures that are found at the centromere. Other microtubules will spread out in a formation called the aster, connecting to the cell membrane and their counterparts from the opposite side of the cell. The microtubules connected to the chromosomes will gradually center them along the metaphase plate. The next and shortest stage of the cell cycle is known as anaphase. During this stage, the microtubules connecting to the chromatids begin to shorten. The chromatids will separate and travel to either end of the cell. As soon as the sister chromatids split, they will become identical chromosomes. As soon as the chromatids separate, the microtubules connected to each other and to the cell membrane will begin to lengthen, which will cause the cell to push away from the metaphase plate. This process is known as telophase. During this phase, the nuclear envelope will also begin to reform around each cluster of chromosomes. The chromosomes themselves will begin to dissociate into their chromatin state. Once the cell has completed telophase, it will commence with cytokinesis. This process is different in plant and animal cells. In animal cells, a cleavage is formed by the creation of a ring of microfilaments along the metaphase plate. As the ring contracts, the cell will be split into 2 genetically identical diploid daughter cells.
Meiosis is a cellular process similar to mitosis in that it results in cell division and contains many of the same steps. However, unlike mitosis, meiosis produces 4 genetically unique haploid daughter cells. Also unlike mitosis, meiosis produces gametes, the cells involved in sexual reproduction. Interphase of meiosis is identical to the process that mitotically dividing cells undergo. The result is a diploid cell with 2 identical sets of DNA. However, prophase 1 of the meiotic cell cycle is very different from its mitotic counterpart. The sister chromatids condense and connect to each other at the centromere. In meiosis, the homologous pairs also connect to each other. Homologous pairs differ from sister chromatids in that chromatids are identical and homologs are not. Homologous pairs are the chromosomes which code for the same genes in an organism. However, one homolog comes from each parent in sexual reproduction. During prophase 1, the chromosomes undergo a process known as crossing over, where the chromosomes trade portions of their DNA with their corresponding homologs at the chiasmata. The remaining Metaphase 1 is similar to mitotic metaphase in that the microtubule connects to the kinetochores of homologous chromosomes and line them up along the metaphase plate. Anaphase 1 is very different from its counterpart because it does not pull apart the sister chromatids. Instead, the microtubules pull the homologous pairs apart, leaving the centromeres of the sister chromatids intact. The selection of chromosomes to each end of the cell is random, so some maternal and paternal homologs will appear in each eventual haploid daughter cell. Telophase 1 of meiosis is similar to its mitotic counterpart, as is cytokinesis. The major difference is that the resulting daughter cells are haploid and genetically unique, but with 2 sets of DNA each. In order to correct the double dose of DNA, the cell must undergo the mitotic cell cycle again. Very little happens in prophase 2 as the DNA is already condensed and the nuclear envelope has not formed. Metaphase 2 is nearly identical to its mitotic counterpart. In anaphase 2, the sister chromatids separate. However, unlike in mitosis, the sister chromatids are not identical as they have retained some of the DNA from their homologue. In telophase 2, the chromosomes decondense and the nuclear envelope forms around them, and cytokinesis begins. The result at the end of meiosis is 4 unique haploid daughter cells.